Hello, my name is Augustus Pendleton. I work in the labs of Dr. Grolnick and Dr. Bond at the University of Minnesota. Today I'm going to tell you honestly, about something really, really boring. So, I'm breathing right now, you're breathing right now, everyone around us is constantly breathing. And what we're really doing when we're breathing is we're taking electrons off of the carbon we eat, which is food. We're taking electrons off the carbon and we're putting them onto the oxygen that we breathe. And then we're getting rid of them. Pretty boring. I breathe all the time, you breathe all the time. We've got the breathing down, which is good. However, I want to talk about some really cool bacteria, especially one called Shewanella. Okay, Shewanella. Shewanella can take the carbon, or the electrons it takes off of carbon, it's food, and put them onto crazy things like iron, like uranium, various sulfur compounds. It can use many different chemical compounds and metals as a terminal electron acceptor. So that's really exciting. And we want to understand more about how they do this, because the truth is that's very, very difficult. When we're moving electrons onto oxygen, that oxygen is inside the cytoplasm of our cells. However, for Shewanella, it's making oxygen in it, or it's making electrons in it's removing electrons from carbon in its cytoplasm. But because it's a gram-negative bacteria, it needs to take those electrons from the cytoplasm where it ripped them off of carbon, cross over an inner membrane, cross a space called the periplasm, and cross over an outer membrane to get to the outside world. And then once it's there, it can interact with the metals that are sitting out there. This is really, really hard. But research has shown, especially like research done in the labs of Dr. Grolnick and Dr. Bond, that there are very complicated and sophisticated pathways of enzymes and proteins that sit on the inner membrane, they sit in the periplasm, they sit on the outer membrane, and they act as this bridge to move electrons across the periplasm and then into the outside world. So that's really exciting. My protein that I want to talk about today is called FCCA. FCCA is called fumarate reductase. It has two jobs. It will reduce fumarate. Okay, it will use fumarate as a terminal electron acceptor. And it can act as a bridge between these inner membrane proteins and these outer membrane proteins to create that larger electron bridge from the cytoplasm to the outside world. However, FCCA isn't used as much as other proteins for this like bridging function, and fumarate isn't really that common in the wild. So it doesn't seem like it'd be that important of a protein. But it's the most abundant protein in Shewanella's periplasm. There's tons and tons of FCCA sitting in Shewanella's periplasm, and we don't know why. We have some ideas, but we don't know why. So my research seeks to find is perhaps not why it is there, but at least how did it get there? So proteins have to be moved across the inner membrane. They're made in the cytoplasm. We have to move them across the inner membrane into the periplasm. So first determining how is FCCA moved into the periplasm. And then also FCCA is a flavoprotein. It has a flavin cofactor. And so that flavin cofactor is FAD. And so understanding how FAD, which is also often made in the cytoplasm, is moved into the periplasm and interacts with FCCA. By understanding how FCCA gets there and becomes functional, we'll hopefully have a better understanding of why it is there taking up so much space for something that doesn't seem that important. And so why do we care about really fine-tuning this bridge between the cytoplasm and the extracellular space? Well, there's a couple of really awesome things you can do with metal-reducing bacteria. For one, uranium in an oxidized form is extremely soluble, and it's also extremely toxic. So if you have some sort of plant that uses uranium and it's releasing wastewater with soluble uranium, that can leach into surrounding waters. However, when Shewanella reduces uranium, it becomes a solid precipitate and falls out of water. So now you can clean up your water before letting it leave the factory without contaminating the outside environment using a biotechnology solution. The other really exciting thing is Shewanella can pass electrons onto graphite electrodes. So what that really means is you can have two dishes of Shewanella culture and they're moving electrons between electrodes, making a current. So you can have a bacterial sourced current going on. And this could be a really amazing way to create sustainable, small systems of constant current for a long time. So even though to me our breathing is sort of boring, all we do is breathe air, I'm really excited to share more about Shewanella's ability to breathe various metals like iron and uranium, and understand why FCCA, what role it has within that system. Thank you so much for listening.